Assalamu alaikum. So today we are starting with chapter number 16, inherited change. And let's go to the learning objectives now. So objective 16.1 talks about passage of information from parents to offspring, right? So it says explain what is meant by homologous pair of chromosomes, explain the meanings of terms haploid and diploid, and the need for reduction division, which is meiosis, prior to fertilization and sexual reproduction. Then explain how crossing over and random assortment of homologous chromosomes during meiosis and random fusion of gametes or fertilization lead to genetic variation, including the expression of rare recessive alleles, right? Then after that, we are going to talk about the role of meiosis and gametogenesis in humans and the formation of pollen grains and embryo sacs in flowering plants. Now, remember that you can be asked to describe, right, with the aid of photomicrographs and diagrams, the behavior of chromosomes in plants and animal cells during meiosis and the associated behavior of the nuclear envelope, right? The cell surface membrane, the spindle, right? In this case, names of main stages are expected, but the subdivisions of prophase, meaning the name of subdivisions of prophase is not included, right? It's not needed. So these are the learning objectives for today, and let's move on to the main topic for meiosis. All right, then let's talk about meiosis. Now, for understanding meiosis, we need to first recap the cell cycle, though you have already learned about that in AS biology, but still a recap is required. So cell cycle is actually a regular sequence of events that takes place between one cell division to the next, right? So when we talk about this entire cell cycle, it consists of three phases, right? And the first phase, this one here, starting from here till here, or around like 80 to 90% constitutes interphase, right? So interphase itself is then divided into three stages. First phase is the G1 stage, which is known as the growth phase. Then we have the S phase of interphase. So this is the phase actually where DNA replication takes place, right? And then G2, which is the cell growth. So the DNA that has been replicated. So if there are any errors there, so they are actually rechecked, and then they are actually like the errors are fixed, right? And after that, the cell undergoes the second stage, which is known as, like right now in this case, we'll be talking about meiosis. That is what we call as a nuclear division, right? And after that will be cytokinesis, and that will be the part when the cell is going to divide, right? So this whole entire cell cycle is made up of three different phases, right? So we have interphase, right, which itself is divided into three phases, G1, S phase, and G2. Then we have the nuclear division. Like nuclear division is the one where either mitosis or meiosis will take place. In this case, we'll be talking about meiosis. And third one is cytokinesis, the one in which the cell is going to divide, right? So a cell remains in the interphase for around 80 to 90%. So major chunk is there in the interphase, right? Mitosis or meiosis constitute about around 10, 20%, right? And the same is the case of cytokinesis, right? So this is how the cell cycle is. And like we have to be actually, we'll talk particularly about the S phase of the interphase. So let's move on to the next part then. So let's clarify the concepts about chromatin, chromosomes, and chromatids as well, because again, like I've seen students getting confused with these terms. So remember that whenever we talk about chromatin, it is this part here. So it is actually the loosely coiled form of chromosomes. Remember, these are all like these are chromosomes. These are simply loosely coiled form of chromosomes. And this is happening like this is during the interphase stage, right? This one, because remember the cell actually remains in interphase like most of the time, right? So it is like this is how the chromatin actually stays if you look at G1, right? So the chromosomes will be in a loosely coiled form, and that is what we call them as a chromatin, right? So this will be during the interphase, right? Chromatin itself is made up of DNA and the proteins, which are known as histones, right? And it is visible as loosely distributed patches of fibers within the nucleus when stained, right? So you will be able to see when the cell is not actually, the nuclear division is not happening during interphase. The cell, if you're looking at the cell, you would find chromatin in this stage. So it's a loosely coiled thread-like structure, right? Now, so remember this is during the interphase, right? Uh, G1, the growth phase, and during DNA synthesis, right, the S phase, it will be simply replicated, but it will stay as a loosely coiled thread-like structure. Now coming to chromosome, again, these chromatids, right? So chromosomes are a structure made of DNA and histones, the same, right? They're found in the nucleus of eukaryotic cells. So remember, these are the same, right? But simply what happens, like chromosomes actually during the prophase, as we will be talking about prophase in meiosis also. But what happens as the nuclear division starts, the chromosomes begin to condense, they become to thicken, and these coils, these loosely coiled chromatid actually starts to condense more. So they actually become super coiled, right? So they become very prominent. So then we call them as a chromosome. So it's exactly the same thing, but now it's simply the supercoiled form of the chromatin. So here, depending on the different stage of the cell cycle, right? We can have a single chromosome, or when the replication is taking place, it can be duplicated chromosome, right? So chromatin, if you're talking about the structural differences between the chromatin and the chromosome, so remember chromatin, the fibers are long and thin. They are uncoiled structures found inside the nucleus. And when you're talking about chromosomes, these are compact, thick, and ribbon-like, right? And these are coiled structures seen prominently during the cell division, right? So this is at different phases of cell cycle, the chromosome will actually look like that, right? Coming to chromatid, sorry. So chromatids, one of the two identical pair of chromosomes. So this is a chromosome, right? And like this one here is a duplicated chromosome. So this is one strand, right? The one you're looking at here. This is one strand of chromosome, right? And this is another one, right? So what we're looking at here is this, one of the two identical parts of a chromosome held together by a centromere, right? So centromere is the circular structure that you're looking at, right? So the centromere actually holds these chromatids together, right? And this is formed during interphase when the DNA is replicating, right? So when the DNA is replicating, this single chromosome, right? Because this is one strand, it's a chromatid, it will divide into two. But when it divides into two chromatids, remember these are known as sister chromatids, and they're joined together by the centromere. So this is the centromere, and both of these two will be known as sister chromatids, right? So these are the basic terminology that you should be familiar with. You should be actually referring to these. So chromatin is the loosely coiled thread-like structures, right? These are again, these are loosely coiled forms of chromosome, but you will find them during the interphase, right? Chromosome, when the same chromatin becomes supercoiled, thick, prominent, becomes condensed during, right? So then it is known as a chromosome, right? And then since like DNA replication takes place during interphase, right? So again, you will be looking at a duplicate chromosome. And these duplicated chromosomes, like they have these two chromatids, which are attached together by a centromere, and both of these together are then known as sister chromatids. Okay. So when we talk about meiosis, it's basically the passage of information from parent to the offspring. So we will be referring to the term homologous chromosomes. Now, homologous, right? Homologous means like it's sort of matching, right? Matching pairs. So they are similar in position, structure, and evolutionary origin, but not necessarily in function, right? So remember, like if you're looking at the length of also, so that the length will be the same. So they will be similar in position, the structure would be the same, right? Evolutionary origin would be the same, but not necessarily in function, right? So what are homologous chromosomes? They are simply two pieces of DNA within a diploid organism. When we're talking about diploid organism, we are talking about 2N, right? A diploid organism that has 46 chromosomes or 23 pair of chromosomes, right? So these homologous chromosomes, right? They are two pieces of DNA within a diploid organism which carry the same genes, right? Remember, they will have the same genes, one from each parental source, one from the mother, one from the father, right? So those two will match. They will form homologous chromosomes, right? One matching will be coming from the father, one from the mother. Both of them together will be known as a homologous chromosome set, right? So now, since we'll be talking about humans here, right? So there are 22 matching pairs of chromosomes, which we call homologous chromosomes. Remember, we are talking about 22 matching pairs, right? And in pairs, One set is coming from father, one is coming from mother, and both of them are homologous. Like both of them make homologous matching. Similar in position, similar in size, similar in structure. Each pair is then given a number, right? Like from 1 to 2, right? In the original zygote, one of each pair came from the mother, one from the father, and we have a non-matching pair also, which is called X, right? And then we have Y also exists. Hence, what we have two sets of 23 chromosomes. Remember, one set is actually haploid, right? And haploid is one set. Okay, when it is diploid, we call it two sets, right? So we have two sets of 23 chromosomes. It means 46, right? One set of 23 from the father. Remember, one set means haploid, right? It is N. So one set means, let me write it here. It means we're talking about haploid, right? And when we talk about two sets, it means then it becomes diploid because one set is coming from father, one is coming from mother, right? And together it then makes 46. So diploid cell is the one that possesses two complete sets of chromosomes, and the abbreviation for diploid is 2N. Remember? So two complete sets, it means one complete set is coming from father, 23 chromosomes. One complete set is coming from mother, 23 chromosomes. Together they make diploid, which is 2N or 46 chromosomes, right? So haploid cell is the one that possesses one complete set of chromosomes. The abbreviation of haploid is N. So I hope it makes sense, right? Okay. Now this is what is actually known as a karyogram. So karyogram here now look at these homologous pair of chromosomes. See, when we say homologous, it means same in length. So you can see the size, right? You can look at the centromere position of the centromere. Then you can actually look at the arms from the centromere to have the same length. They look same, they have the same structure, right? So you can look at these again, matching one from father, one from mother. Then you can look here. So remember they have been numbered 1, 2, 3, right? So you can look here and see with each subsequent, you might also be noticing that the centromere is actually coming like a bit above, right? It's not as centralized as such as in these cases, right? And then you can see the size is also getting smaller and smaller, but all of these still remain as homologous matching pairs, right? Now, one of them, the one that is here, is actually what is known as, these are actually one set of, we have this sex chromosomes, right? Now this one is for a male because we have one X chromosome and one Y chromosome, right? If it was a female, we had two X chromosomes here, right? So this is what is known as a karyogram. So it shows you these chromosomes, and generally all the autosomes, all the like 22 pairs of chromosomes, homologous chromosomes are shown like this, while the sex chromosomes are actually like shown here at the bottom here, right? So this here, the one that you're looking at here is right now of a male. Okay. So when we talk about the passage of information from parent to offspring, so just understand that a life cycle of an animal, such as human, it consists of two main stages. The first stage is known as the growth phase, right? And growth phase, remember, we have already learned about mitosis in AS biology, right? So mitosis is when the cells actually like the nuclear division happens, but we have the same identical number of cells, right? Or identical number of chromosomes as are there in the parent cell, right? So it has involves growth. And then the second stage of the life is sexual reproduction. So right now, as we are talking about meiosis, we are dealing with this aspect of a human or an animal life cycle, right? So for a life cycle to contain sexual reproduction, there must be a point before fertilization. Remember, it has to be before fertilization. Fertilization means fusion of males and females gamete, right? So there has to be a point before fertilization, right? When the number of chromosomes is halved. So remember, we are talking about diploid cell, right? So at the start, we will have the diploid cell, but eventually this number has to be halved. It means it has to be converted to haploid cell, right? But this has to happen only when the sexual reproduction has to take place. If this was not the case, right, if it was any other cell of the body, right, so then mitosis would continue, right? But this is happening only if sexual reproduction has to sustain, and this has to be happened before fertilization, right? So what happens in that, as this diploid, right, cell converts into a haploid cell, right, or the chromosomes number is half, this results in gametes containing only one set of chromosomes. Remember, when we talk about one set, we are talking about 23 chromosomes, right? I'm not saying 23 pairs. Okay, 23 chromosomes, right? Now, rather than two sets, right? Two sets is actually diploid. Or two sets, may you can use the term, yes, 23 pairs of chromosome, that would make 46, right? So I hope it's making sense now. If there were no point in the life cycle when the number of chromosome halved, then the number of chromosome would double every generation, right? Because if we kept it like 2n, so it would keep on doubling and doubling. So we do not want that. So meiosis is a type of nuclear division. Remember, when we talk about cell cycle has three stages: interphase, nuclear division, and then comes the cytokinesis. So we are talking about nuclear division, right? So it happens after the interphase. So it's a type of nuclear division which does this, and hence it is also called the reduction division. Reduction because it is going from 2n to n, haploid. So again, just for your understanding, that you need to know the difference between diploid versus haploid. So remember, diploid is always two sets of chromosome, right? We're talking about this. One strand is known as one set of chromosome. Remember, right? This is haploid. When we're talking about diploid, we're talking about two sets. So this is one set, this is another set. So this is 2n, right? 2n or two sets of chromosomes. Okay? And since we're talking about meiosis, so they will be made into one set. So starting from 2n, right? We will have these haploid number of cells. We will have one set. So if you're talking about examples of diploid cells, almost all cells of our body are diploid. Most plants and animal cells are diploid, right? But when we talk about haploid, so again, these are the like cells which are found in the reproductive organs, right? So these are the gametes. Okay, so female gamete, egg or ovum, and male gamete is sperm. They are haploid. And remember, as we were talking about, this has to happen before fertilization, because once fertilization happen, right, the male gamete and the female gamete fuse. So 23, one set from the father, 23, one set from the mother, they fuse, and then again we have those 46 chromosome. It becomes the zygote that becomes diploid. I hope you're getting it now. Now remember, whenever we are talking about the parent cell is the cell like actually which would undergo mitosis or meiosis, right? So the parent cell, which is at the start of the cell cycle, is always diploid. Remember, this is one similarity between mitosis as well as meiosis. The parent cell, the cell that starts to divide, will always have diploid number of chromosomes or 46 chromosomes, or we may say it has two sets of chromosomes, right? So at the start of all the cell cycles, whether it's meiosis or it's mitosis, the parent cell will be diploid, right? Now, the thing is when we say 2n, right, it simply means quantity of DNA within the cell. Quantity of DNA, right? That is actually what we refer to because we're talking about the number of chromosomes here, right? We're talking about two sets of chromosomes. Two sets means we are talking about 46 chromosomes, right? So that refers to the quantity of DNA within the cell. Now, here they have shown you those homologous chromosomes, right? So this is actually what they've shown you. This one is coming from the father, right? This is coming from the mother. And remember, this is one set, haploid, right? This is another set. So this is actually something you see, it's like before fertilization, right? So although the paternal chromosome and the maternal chromosome are of similar length, remember we're talking about homologous, similar length and contain the same genes at the same loci, they may contain different alleles. Now alleles are different variations of a gene, right? For example, the gene may be of eye color, right? But like for example, capital B might be actually coding for brown eyes, blue might be coding for black eyes, right? Still, it's like the gene remains for eye, but allele, one is for coding for brown, other is coding for black, right? So alleles might be different, but again, when we're talking about homologous, so there will be the matching pairs. One set is coming from mother, one is coming from father, but the matching ones actually pair up. This is what makes the homologous chromosome, right? And the genes would be in the same loci. So you're looking at loci means the same positioning, the position at which the genes are located. So here you can see loci or loci, like so you can see this B and this B, same positioning, okay, on two different chromosomes, and D on two different chromosomes, right? So these are the same, like alleles, capital D, capital D. These are two different alleles, capital B and small b, right? Right. So just remember when we talk about homologous chromosomes in diploid cells, right, they would come in pairs, right? So this is what they've shown you here. Now, so we were talking about the parent cell at the beginning of the cell cycle will always be diploid, right? So they're asking you can name the stage of the cell cycle that occurs before the cell division. Remember, we're talking about before cell division, meiosis, and state the key event that occurs during this stage. So remember, we're talking about like the stage which happens before cell division. So it has to be interphase, and DNA replication takes place during interphase, right? Or the S phase, right? So this is the part of the meiosis, like, because this stage happens before meiosis starts, right? DNA replication occurs, right? So homologous chromosomes, they would replicate, right, to form the sister chromatids. So remember, we had the diploid cell, right? It had 46 chromosomes, right? But then as it replicates, so now we have 92 chromosomes because it replicates, everything doubles. So homologous chromosomes, they replicate to form pair of sister chromatids. So here you're looking at this one, right? Like if we say the sun is coming from the father, it says blue, right? So this was B, capital B and capital D here. So if it replicates, it's the exact same copy. So you can see a capital B here and a capital D here. So exact same copy, but these sister chromatids are now attached by centromere, right? So centromere actually is the attachment site of these two chromatids. So remember, this is one chromatid, this is another chromatid. Together we are calling them as chromosome. And one chromatid here contains one DNA molecule. This is one complete DNA molecule. This is another complete DNA molecule. So since DNA has replicated, so this DNA molecule has replicated, right? So exactly same, identical copy. The same, if you have a look at here, let's say this is for mom, right? So look at this, B, small b, capital D, it has replicated. So see, that's exactly the same copy, right? So these are connected by centromere, and we call these chromatids as sister chromatids. So sister chromatids, they are attached through the centromere, right? So the centromere is actually a specialized sequence of DNA, right? But there is no genetic material present here, right? It actually just binds these two sister chromatids together. Right. So sister chromatids are genetically identical. Remember, is exact same copy. Non-sister chromatids are chromatids of homologous chromosomes that are not identical, may contain different alleles at the same gene loci. So understand this point here. For example, if we know okay, these two sister chromatids are joined together by centromere, but what about this strand and this strand, right? Let me use pen here now. See, this part here and this part here, right? Because we are saying they are homologous, right? If they are close together. So this strand, this chromatid and this chromatid, since this chromatid is a part of one chromosome, this is a part of the maternal side, right? So if they are connected, they are actually close together. So we call them as non-sister chromatids. Let me use a different pen layer. Yeah, so we'll call them as non-sister chromatids. So still they are chromatids, right? But we'll call them as non-sister, right? So when we talk about genetic variation, so it is actually these non-sister chromatids that are involved there, right? So I hope it makes sense now. Sister chromatids are actually joined together by the centromere. Non-sister chromatids are not joined together by centromere. These are two strands, one from the mother side, one from the father side, right? But these are not connected through the centromere, right? So it is easy to distinguish between sister and non-sister chromatids because later are not joined by centromere. So we just saw that, right? But non-sister chromatids, remember, they play a very important role in genetic variation, and we'll see that in a while, right? So these are the non-sister chromatids. Now, since we were saying that the DNA replicates, so remember DNA replicates during the S phase of interphase, and remember this whole cell cycle, when it starts, the cell is in a diploid state. It's 2N. But as the DNA replicates from 2N, it converts into 4N just before meiosis starts, right? So remember, when meiosis starts, because the DNA has already replicated, the DNA will be 4N. So from the 46 number of chromosomes that we had initially in the diploid state, right? after interphase, as the DNA has replicated, we are talking about 92 chromosomes here, right? Now, so the process of meiosis, remember, now it is different from mitosis in the sense that we have two nuclear divisions there. In mitosis, we had only one nuclear division, right? So we have two nuclear divisions here. There is usually in the formation of 4 haploid daughter cells. Remember, we talked about we'll start from diploid, but eventually we will have haploid cells. So 2 nuclear divisions takes place usually, and finally we have these 4 haploid daughter cells from a single diploid parent cell. Genetically different daughter cells form from the independent assortment of homologous chromosome. So these are the key terms that we will be talking about as we start discussing meiosis. We will again use these terms and then I'll talk about in detail of what these terms actually mean. But remember, these are like special events that take place during meiosis. So genetically different daughter cells come from the independent assortment. Assortment, take it as a combination. So we have different combinations of homologous chromosomes. If there's a different combination, we will definitely have genetically different daughter cells. Unlike mitosis, where we were having genetically identical daughter cells, right? And one more is a process that will take place is crossing over. So remember, we were talking about non-sister chromatids a while back, right? So crossing over between homologous chromosomes results in further genetic variation among the daughter cells. So we will see how this crossing over and how this independent assortment takes place as we talk about meiosis in detail, right? So meiosis produces daughter cells that are genetically unidentical to each other and to the diploid parent cell. So even they would not look the same, like आपस में भी एक जैसे नहीं लगेंगे and they're different from the parents also, right? So 2 nuclear division of meiosis, they result in formation of 4 haploid daughter cells. Please remember they're genetically different to each other, right? Now, there is another similarity when we talk about mitosis and meiosis. One similarity that we talked about, yes, we always start with the diploid parent cell, 2N, right? But another similarity in terms of the phases, right? Remember, meiosis, we just said there will be 2 nuclear divisions, unlike mitosis. Mitosis had only 1 nuclear division, but the names of the phases will remain the same, right? In meiosis, if you remember, we had this mnemonic, we had P mat or purple mat, okay? But the names of the phases will remain the same. But what we do in meiosis, we just like assign Roman numerals to them, right? So in meiosis 1 and meiosis 2, right? So in meiosis 1, the phases are P stands for prophase, right? But since we're talking about meiosis 1, we call it prophase 1. Then we will have metaphase 1, then anaphase 1, and telophase 1. And as we talk about meiosis 2, so it will be prophase 2, metaphase 2, anaphase 2, and telophase 2. So again, a sequence that you need to remember, right? So it's simply P mat, right? Or purple mat, right? And remember, interphase will be before that. But remember, interphase is not a part of nuclear division, right? It's a cell cycle. First stage of cell cycle is interphase, when the cell growth happens, right? Then the like DNA replication happens during the S phase. Then we have G2. When if there is any error in the DNA replication, those are corrected, right? And then we will have the nuclear division, which starts from prophase, right? So again, there's another way you can just remember them. Prophase, just think about prominent, right? Because this is the phase when the chromosomes will become prominent. So we'll see that in a little while. So just remember from P, prophase P, prominent. Metaphase M, you can actually remember it from middle, right? Anaphase, you can remember it from maybe apart or away, right? And telophase, you can simply remember it as 2, right? All right, then let's talk about meiosis 1, the first nuclear division, right? Prophase. Now, when we talk about prophase, so this event occurs and in meiosis 1, right? We're talking about prophase 1, but remember it is like very similar to what happens in mitosis, right? So, and most of the activity actually takes place during prophase, right? The chromosomes, right? Because they have already duplicated in the S phase of interphase, right? So they would now become more prominent, right? The chromosomes would condense, it would thicken, it would become more prominent now, right? So just remember, prominent starting with the P, prophase is starting with the P, right? And what is happening? The chromosomes, they shorten and they thicken as well as they condense, right? So here you can look at, here they have shown you these chromosomes, like 2 pairs they've shown you, right? So they become thickened, they shorten and they condense. And what happens now? Pair of homologous chromosomes would line up closely together. So the homologous chromosomes, which are of the same like size and structure, one from father, one from mother. You can look at the color coding here also, blue and red, right? So they would now line up close together, right? What also happens, the centrosomes, because they have also replicated during the S phase, when the centrosomes move towards the opposite poles of the cells. So make sure you're using the term poles. Please do not use the term sides of the cells, right? It's this structure is not a regular like square or rectangular structure. So make sure you're using the term opposite poles of the cells, right? And at the same time, the nuclear membrane also starts to disintegrate. So you can see this is like how it starts. These are the centrosomes, and they will start to move towards the opposite pole, right? The homologous chromosomes, they pair up, right? And you can see the nuclear membrane actually has started to disintegrate. So this nuclear membrane is made of the same phospholipid bilayer as our cell membrane. Right? So this will disintegrate, right? The contents will remain there in the cell. And once it regenerates at the end of this cycle, right? So the same contents will be then utilized to regenerate the nuclear membrane, right? So the thing is, you're not like required to remember the names like this. You get early prophase, middle prophase, late prophase, right? That this is not a requirement of your syllabus. You simply just remember it as कि prophase में ये सारी चीजें हो रही हैं, right? So prophase में, see what was happening? The chromosomes become condensed, they thicken, right? They become more prominent. And what happens next? The homologous chromosomes. Homologous, again, I'm keeping on emphasizing the fact they are the same structure, same length, right? Coming from mother and coming from father, right? So they would pair up. This process of pairing up is known as synapses. So again, a keyword that definitely examiner will be looking for in your paper. So the process as a pair up is known as synapses. And at this stage, each pair is called a bivalent, right? So this is one pair, this is another pair, right? We will call them bivalent, right? And bivalent means because both of them have two chromatids each, and like if you like talk about them like separately as a separate chromatid, so these are 4 chromatids, so then we call them as tetrad, right? So just a way as like you're referring to them, right? So if you're talking about them as pairs, right? So we will call them as bivalents. And when you're talking about the 4 chromatids separately, like 1, 2, 3, 4, then we call them as tetrads, right? At the same time, the centrosomes are moving to the opposite. Again, make sure you use the term opposite poles, right? And then the nuclear membrane also breaks up, right? Now, at this stage, crossing over of chromatids may occur. Now, we just used the term crossing over a while back. So crossing over, let me just tell you a small bit about it now, but we'll talk about that in detail also. So crossing over is this stage. Remember we talked about it happens between the non-sister chromatids. This blue is one sister chromatid. This red is one sister chromatid, right? But if you're talking about these strands here, right? This blue strand coming from the father, this red chromatid coming from the mother, but these are non-sister chromatids because they're not connected by the centromere. See, this is the centromere. Centromeres join the sister chromatids, right? So these are the non-sister chromatids. So it's crossing over what happens, that in generally it happens like down the length, below the centromere somewhere, and like these are the longer length of these chromatids here, right? So these bivalents, as they have paired up, remember homologous chromosomes pair up, bivalents, right? They show crossing over chromatids. What happens because of the crossing over? Now they would cross over, right? And chromatids may then break and they may reconnect to another chromatid. But this breaking and reconnecting would take till anaphase, right? So they will remain in this stage, right? Because this is happening during prophase. They will remain the same during metaphase. Okay? It only during anaphase when the chromosomes are being pulled apart, right? Only in that phase they will break, right? But they will stay disconnected in this stage throughout prophase, metaphase, but they will break during anaphase, right? So this point where actually this crossing over is taking place. So you can see the red one and the blue one. This point here is known as chiasma, right? So it's a singular, we call it as a chiasma, right? And as a plural, we call it as a chiasmata because this crossing over may take place here also. Right? It can take place in more than one places, right? So then if it's more than one place, a plural, we call it as a chiasmata, right? One or more chiasmata may form anywhere along the length. So remember, this happens along the length, but it happens only between the non-sister chromatids, right? So crossing over is one special event that happens in prophase 1, right? So just have a look at it one more time. Chromosomes shorten, thicken, and condense. The part of the homologous chromosome would line up together, and the lining up or the pairing up is known as synapses, right? So as a pair, we call them bivalent, or if you're talking about them individually, we use the term tetrade, right? Centromeres, centrosomes also move towards the opposite ends, right? What happens next? The nuclear membrane starts to disintegrate, right? As well as crossing over may also takes place during this stage, right? So crossing over is actually the part where the genetic material would be exchanged. And this is the part, as we talk about, the genetic variation takes place during meiosis, right? So this is what happens during meiosis 1. And lastly, as the centrosomes were moving towards the opposite ends, opposite poles of the cell, right? At this stage, they would actually start forming a spindle also, right? So spindle is also formed by the end of prophase, right? So here, this is what they've shown you as a crossing over. So these are the non-sister chromatids, and you can see it happens along the length, right? So because these chromatids are longer, these arms are shorter. So if these arms are actually longer, right? So there are chances of crossing over more in this case, right? So this part here is known as a chiasma. So they've shown you only one part where they're crossing over. So this part is known as chiasma, and this is what actually will lead to genetic variation, right? So this is one event which is very unique to prophase 1. Remember, so you need to know it happens in prophase 1. So make sure if you're talking about your specificity of your answers, right? So do not use prophase only, right? It's prophase 1, right? It is known as crossing over. It allows for the exchange of genetic material to take place. And we have already talked about during crossing over, structure called chiasma randomly forms, creates a point of contact between chromatids. This allows for exchange of genetic material, right? And the chiasma forms between non-sister chromatids, as these are not identical. Therefore, the exchange of genetic material can lead to new combinations of alleles that were not seen in the parents. And remember one more thing about these chiasmas, right? They would never ever form in a point कि कोई gene disrupt हो रहा हो, ठीक है? So ऐसा होगा कि for example if they're being formed, so gene या तो उसके नीचे है, ठीक है? कि वो complete जो genetic variation होगी वो complete होगी, ये नहीं कि वो किसी चीज के बीच में आ जाएगा तो वो gene वहाँ पे वो डैमेज हो जाए, ठीक है? तो वो ऐसे किसी particular point पे अटैच होगा कि gene would be below it, ठीक है? ताकि जगह क्रॉसिंग ओवर हो रही है तो this whole strand actually gets exchanged, right? So what they've shown you here, they have shown you one chromosome from the mother and one from the father, and they have shown you this homologous pair. So they are saying that these three recessive alleles are coming from the mother, right? A, B, C. The recessive is small a, B, C. The small letters we generally refer to as recessive, and capital letters we refer to as dominant, right? So they're saying this one is coming from the mother, this one is coming from the father, and see both of them are homologous, right? So, okay, so remember what will happen. So this is the 2n stage, right? Because this is one set from the mother, this is one set from the father, right? So they will make a diploid cell, which is 2n. But remember, during interphase, DNA replication was taking place. So this will also duplicate, right? And the father's chromatid will also duplicate, right? So these are now, we have these homologous pair of chromosomes. The one that which we will call is bivalent, right? Since there are two pairs, 1, 2, right? And 1, 2, 3, 4. If you're talking about this like this, we'll call them as tetrads, right? So as a result of DNA replication during the interphase, each chromosome duplicates, and it is duplicating to see the exact same copies or exact same copies are made here, right? For mother as well and for the father as well, right? So they have identical alleles. Remember, alleles are versions of this gene. So here they have the same alleles, exactly same, right? So we have now sister chromatids. Now, if we think that this crossing over does not take place, and remember when we talk about crossing over, it takes place between the non-sister chromatids. Here, what they've done, they've not shown you the centromere, but remember these are two sister chromatids, right? And these are two sister chromatids, right? These and these are non-sister chromatids. Crossing over takes place between the non-sister chromatids. Remember, so if we assume that crossing over did not take place at the end of mitosis, at the end of meiosis, sorry. What do you think? This whole, these whole genes will be inherited by the, like, by the child, right? So they would have, if you think about it, they would have 50% coming from the mother, 50% coming from the father, right? So 50% of the, like, they will have this ABC, and 50% would have capital ABC, right? So कोई exchange material, genetic material का हुआ ही नहीं है, ठीक है? So they would be passed along as such. But if crossing over happens, okay, between the non-sister chromatids, now there are more chances of exchange of genetic material at this stage. So see, remember I told you कि it happens, ऐसा नहीं है कि बीच में यहाँ पे कोई allele या gene disrupt होगा. So see, this crossing over is taking place, ठीक है? ये वाला allele ऊपर है, nothing happens to this one, and these two are down below. तो जब exchange होगा, so these two will be exchanged together, right? तो ऐसा नहीं होगा कि वो disruption हो रही है. So see here, this is how now this will be exchanged. So what will happen? What will happen is the chiasma formed between the two non-sister chromatids is the point which lies between the loci of A and B genes. Okay, we are talking about capital A and capital B. So this is capital A, this is capital B. इनके बीच में हो रहा है, ठीक है? तो कोई allele बीच में disturb नहीं हो रहा है, ठीक है? It allows for the exchange of genetic material. See? So what is happening? The formation of chiasma is completely random. Remember, it is random, right? It can happen along the length, okay, of the chromosome, right? For this reason, chiasma can form at any point along the length between the two non-sister chromatids, right? And if crossing over exists, see, these are the non-sister chromatids. So now look here, right? Remember, these capital B and capital C were a part of this paternal chromosome, right? But now since after crossing over, they have been exchanged. So now you can see genetic variation here. This one was a maternal, small a, and now we have a small b and a small c here. So this is what we have now. Genetic combination have been made here. So what would happen? Like these will be inherited as such because nothing happened here. This will be inherited as such, nothing happened here. But if you talk about these, right? So at the end of the meiosis, what would happen? 25% of the gametes contain chromosomes with the allele ABC. So remember, these are 4 now. So 25% contain the same from the maternal side. 25% contains the same capital ABC from the paternal, from the father, right? But if you talk about these, okay, collectively 50 will have this genetic variation, but if we talk about them individually, one would be have a small a, capital B and capital C, that would be 25%, and the other 25% would be capital A, small b and small c, right? So we have these genetic variations now, and this happens because of crossing over, which only happens during prophase 1 of meiosis 1. Right? So remember, as I talked, I told you a while back, this crossing over, this will remain, they will remain in the same state till anaphase, right? Anaphase, or like you can remember it from away. So this will be the phase when these will be pulled apart, right? So during anaphase 1, the chiasma breaks and the chromosomes are separated, right? But during prophase, during the entire metaphase, right? And they will only separate during anaphase. So remember that, right? And we have talked about, yes, okay, at the end of meiosis, 25% of gametes contain the small letter, the recessive A, and then capital B, capital C. Another 25% contains capital A, small b, and small c. So these are the recombinants now. Okay, यहाँ पे genetic variation हो गई, right? So these two combinations, now we have these two combinations, small a, capital B, capital C, capital A, small b, small c, right? We use the term recombinants for them because these are combinations. Okay, we use the term recombinants for them, right? Because they are combinations which are different to those of the parents and only arise because of crossing over. So जो भी variation crossing over की वजह से different combinations बन रही हैं, we use the term recombinants for them. We call them as, like we use the term कि genetic recombination हुई है, ठीक है? We will simply refer to them as recombinants. So remember, they are different from those of their parents and they only arise because of crossing over, right? So crossing over always, like it occurs during prophase 1. So please remember, prophase 1 results in recombinants. Okay? So these are reassortments. Assortment, again, think of it as a combination. So these are reassortment of alleles. So जो alleles already पहले से थे, अब उनकी reassortment हो गई, वो दोबारा से एक different new तरीके से combine हो गए. Okay, so these are reassortments of alleles into combinations that are different to those of the parents. I hope it makes sense. Coming to the second one. So this is metaphase 1. So remember metaphase M, remember it from middle, right? So what is happening during metaphase 1? Now remember, crossing over, this will still stay there. So see, crossing over khatam nahi hai, it stays there. Sahi? So bivalents now line up across the equator. Acha, we use the term middle, okay? But middle as such, middle सिर्फ याद रखने के, but generally this is actually the equator of the spindle. So make sure you're using the term equator. So bivalents, they line up across the equator of the spindle, and they're attached by the centromere. So you can see these are attached by the centromeres, right? And they will be actually, they will line up. Because they seem to line up in the middle, but make sure you're not using the term middle, you're using the term across the equator of the spindle, right? And spindle formation, so you can see these are the centrosomes, right? So they have actually made these spindle fibers, and now these bivalents would just start to attach there. They would line up, right? So during metaphase 1, pair of homologous chromosomes align at the metaphase plate. So we use the term metaphase plate for it as well, right? Or we use the term equator of the spindle, right? And this is also completely random, right? Completely random means the blue could be over on this side, the red over could be over on this side. Okay, so it's completely random, right? Or maybe this blue and this red, then maybe they would actually like uh, align over here and these two align over there. Okay, so we use the term actually independent assortment for that also. Okay, so it actually it's up to them to how they arrange. जरूरी नहीं है कि ये दोनों नीचे ही लगेंगे, ये दोनों ऊपर ही लगेंगे. ठीक है, so it can be anywhere on. ठीक है, ऐसा भी हो सकता है ये flip हो जाए, मतलब घूम के this blue one, ये वाला जो arm है इधर आके attach हो जाए, ये वाला इधर attach हो जाए. ठीक है, so it's completely random. The red one over there, the red one, the blue one over there. ठीक है, so random assortment actually takes place during metaphase, right? This means that on some occasions, the paternal chromosome would be facing a particular pole of the cell, and on other occasion, it will be the maternal chromosome that faces that pole. So I hope you understand, right? We were talking about poles of the cells, right? So maternal or paternal, again, it depends. Just as I was saying, कि अगर ये maternal है, तो it can be on this side, the blue one can be on this side, right? Both of them can be here at the top. These two can be here at the bottom. Okay, so there's totally random alignment, right? Then comes anaphase, right? So anaphase, again, just remember it from away. So this is the time when these will be pulled apart, right? So remember, since we had those bivalents, right? And so what will happen, the centromeres will never divide. Okay? In mitosis, this was happening, but in meiosis 1, right? What will happen? These were bivalents, remember, right? But the centromeres will not, centromeres, but they're with the sister chromatids, right? So this was a sister chromatid. This is one sister chromatid, right? The centromeres will not divide, right? But whole chromosomes, the whole chromosomes, remember the duplicated ones, they will move towards the opposite of the spindle. And generally what will happen as the spindles, they're pulling them, right? So centromere will be pulled first. So you can look at the picture here also, right? So now these are pulled here, these are pulled here. And now it is at this part, then the chiasma that was formed during crossing over, it will break now. So this happens during anaphase 1, right? So they're being pulled towards the opposite ends of the spindle, right? And centromeres first, pulled by microtubules. So these contractions, actually, these microtubules are contracting and they're pulling these chromosomes, right? Centromeres first and then the chromosomes. So these two will move here and these two will move here. And then you can look at this genetic variations here now that will take place because of crossing over. So the random assortment of chromosome during metaphase 1, as I just told you, is completely random. That where there's a which pole towards which pole the maternal chromosome aligns towards which pole the paternal chromosome aligns, right? So it's completely random, and it also results in genetically different gametes, right? So we were talking about crossing over. ठीक है, क्रॉसिंग ओवर के अंदर भी जेनेटिक वेरिएशन हो गई है, फिर इनकी इस तरह रैंडम अलाइनमेंट की वजह से और जेनेटिक वेरिएशन हो गई है, right? So this happens during metaphase 1. So now, okay, we will have genetically different gametes. Okay, coming to the last part of meiosis 1, which is telophase. So again, just remember it from 2. So this is the part अब जहाँ पे वो 2 में divide हो जाएगा. Okay, finally what will happen, because the अब now the nuclear membrane that got degenerated during prophase, अब वो दोबारा से regenerate होगी, बनेगी दोबारा. So once the nuclear membranes reform around each set of chromosomes, क्योंकि chromosomes जो थे, if you look here, ठीक है, they have moved towards this end. These two have moved toward this end. So now what will happen? Now अब इनके around nuclear membranes reform होंगी, ठीक है? So what happens once the nuclear membranes reform around each set of chromosomes? Remember each set. Cytokinesis occurs. Now the cytoplasm will actually start to separate. Okay? And finally the cell will actually, we will have two cells from one cell. Now the cytokinesis occurs where the cytoplasm cleaves and results in two cells which are both 2N diploid. Remember we started with diploid, but interface के अंदर जब DNA replication हुई थी तो वो 4N हो गए थे. ठीक है, अब दोबारा जब ये फाइनली टीलोफेज के बाद अगेन जब वो 2 सेल में डिवाइड हो रहे हैं तो हर एक के अंदर दोनों के अंदर 2N 2N. ठीक है, तो वी स्टार्टेड विद डिप्लॉइड 2N से वो 4N में कन्वर्ट हुआ. ठीक है, मेयोसिस स्टार्ट होने से पहले, प्रोफेज से पहले, उसके बाद दोबारा प्रोफेज, जब प्रोफेज के बाद मेटाफेज, एनाफेज, टीलोफेज तक व्हाट विल हैपन एज द वन सेल पेरेंट सेल डिवाइड इनटू 2, अब जो डॉटर सेल्स बने दे ऑल हैव 2N. ठीक है, so this is what happens during meiosis 1, right? So look at the picture also. So during telophase you can see कि यहाँ पे, ठीक है, एक set of chromosomes, ठीक है, ये दूसरा, and what is happening here? Now the nuclear membrane has regenerated, nucleolus is also reforming, right? And you can see the cytoplasm actually, a cleavage actually forms there. It pinches inward, ठीक है, which we call as cytokinesis, and eventually this membrane or this furrow will actually join together. So we will have two different cells, right? The spindle actually, it will also start to like degenerate here. These are the remains of a spindle there. Chromosomes have reached poles of the spindle. Animal cells usually divide before entering meiosis 2. ठीक है, so animal cells generally divide हो जाते हैं. ठीक है, so we have two cells here. Plant cells के अंदर क्या होता है? They go straight into meiosis 2. ठीक है, with no reformation of nuclear envelopes or nucleoli. So we will see अभी हम आगे जब gametogenesis भी देखेंगे plants के, so you see there. Right, so plant cells, what happens? There is no reformation of nuclear envelopes and nucleoli. Right, so जैसे animal cells के अंदर nuclear envelope बन जाता है. Plant cells में doesn't happen. So during meiosis 2, chromatids separate as in mitosis. ठीक है, so meiosis 2 जो होगी ना, it's generally quite a lot as mitosis. So अभी you'll see as we go through the stages. But this is telophase, and now it has divided into two cells. And remember, these are 2N each. So we have one set here, and take it 2N. And again, we have one set here and one set here, 2N. But see, genetic variation has taken place because of crossing over here. ठीक है, और यहाँ पे ये क्यों हुआ है? Because of random assortment. जब metaphase के अंदर they were aligning themselves. ठीक है, so genetic variation has taken place. Now, this is very important that you realize that although we are talking about two nuclear divisions in meiosis, remember we talked about meiosis 1, and now we'll be talking about meiosis 2. And definitely two sets of cytoplasmic divisions are taking place during cytokinesis, because meiosis 1 के अंदर भी cytokinesis हुई है, ठीक है, और meiosis 2 के अंदर भी होगी. But please remember that interphase only occurs once at the beginning of the cell cycle. So, ठीक है, cell cycle के जो interphase वाली stage है, which is around 80 to 90%, right? The G1 phase, then the DNA replication phase, and the G2 phase. Interface सिर्फ एक ही जगह होती है. Meiosis 1 शुरू होने से पहले जो interface होगी, that will be the only time it will happen. Meiosis 2 से पहले दोबारा interface नहीं होगी. Please remember that. Right? So interface only occurs once at the beginning of the cell cycle. It wouldn't make sense for a second interface, क्योंकि then again just think about it. Interface में क्या हो रहा था? DNA replicate हो रहा था. अगर वही DNA replicate होकर 4N में convert हुआ और फिर meiosis 1 के आखिर में वो 2N में हो गया, तो वो दोबारा 4N हो जाएगा. What's the point there? Right? Because we're talking about this meiosis as a reduction division. ठीक है? And if you think about meiosis 1 was actually a reduction division. 4N से हम लोग 2N पर आए. ठीक है? 4N से 2N पर आए, तो reduction division हुई है. ठीक है? So it wouldn't make for second interface to occur between meiosis 1 and 2, because it would increase the quantity of DNA back up to 4N. ठीक है? When this form of cell division is trying to finish with N haploid. So remember, so this is very, very important that you realize this fact. Interface will only occur at the beginning of the cell cycle. ठीक है? और यहाँ पे जो हम nuclear division की बात कर रहे हैं, वो 2 दफा हो रही है, meiosis 1 and meiosis 2. We have already talked about meiosis 1. We started with 4n and now we have 2n diploid cells. Okay, so let's talk about meiosis 2. Meiosis 2, again, the names will be in the same sequence as PMAT, right? Purple mat or PMAT, prophase, metaphase, anaphase, telophase, but now we'll use a Roman numeral 2 with them, right? So again, the stages will be like almost the same. Prophase, just again think of it as prominent. Okay, so remember, we had these 2n now diploid, right? So what will happen? The centrosomes would then start moving towards the opposite ends, right? The nuclear envelope and the nucleolus disperse. So you can see the nucleolus has like disappeared and the nuclear membrane is also degenerating, right? Centrosomes and centrioles, they replicate. They move to the opposite poles of the cell. But now in this case, like as you go to the metaphase, up here in this case, just spindle formation over here, it is at 90 degrees to what it was formed earlier. Okay, I'll show you the picture. So you focus on this part here. Okay, and I'll show you the metaphase 1 so that you can actually relate well to it. See, this was in metaphase 1. These were the centromeres, and you look at this spindle formation here, right? So these centrosomes, right? And now look at metaphase 2. See, look at the centrosomes here now. Okay, so this is just one thing happened. It actually arranges itself with 90-degree angle. Okay, whatever it was there in metaphase 1, so uske 90-degree difference se banta hai. Okay, so chromosomes again metaphase mein you know that they would line up, okay, across the equator of the spindle. So they would line up separately across equator of the spindle. Okay, so meiosis 1 mein, if you can again, if you want to compare, remember meiosis 1 mein we had bivalence and we had this crossing over taking place. Prophase 2 mein there is no such thing. प्रोफेज 2 में ऐसा कोई चीज नहीं है. See, ये already crossing over की वजह से यहाँ पे you can see this recombinant there, right? But nothing like that would happen in prophase 2. It only happened during prophase 1. ठीक है, तो यहाँ पे prophase 2 में basically क्या हो रहा है? They become the same. They condense, they thicken, ठीक है, they become prominent. The nucleus disappears. The nuclear membrane starts to degenerate. The centrosomes start to move towards the opposite ends, opposite poles, right, of the cell. And metaphase में they start to align themselves. So you can see. Okay, so you can see for this, right? And in the anaphase, again, the same, they will be pulled apart, right? Again, centromeres first. The centromeres then divide and spindle microtubules pull the chromatids towards the opposite pole. In this case, okay, so you can see them, they are being pulled apart because यहाँ पे भी अभी तक वो sister chromatids की form में थे. ठीक है, अब ये sister chromatids separate हुए हैं. So you can see that one is going towards this end, other is going towards this hand. Okay, so you can see, right? And then telophase mein again they are dividing into 2. So you can see the nuclear membrane has been regenerated. Yes, spindle formation fiber jo hai, they are degenerating now. Okay, you can see this cleavage formation, right? And this is just like the telophase of mitosis, but in meiosis, telophase 2, you can see we have 4 haploid daughter cells. So we have 4 cells now. So it's 1, it's 2, then we have 3, and then we have 4, right? So we have 4 cells here now in meiosis 2, right? So we started with the diploid 2n, okay? वो replicate होके 4n हुआ, 4n से meiosis 1 के end पे वो 2n हो गया, 2n से we have ended up in having haploid cells, ठीक है? N. So each will have 1 set of chromosomes, right? Right? So again, the same random assortment as it was taking place in metaphase 1, the same will happen during metaphase 2. So they will actually align randomly. So the same way, right? So if you look at metaphase 1, let me use a laser pointer here. So see, ठीक है, it's totally random. ठीक है, ऐसा हो सकता है ये वाला red ऊपर आ जाता, ये blue नीचे आ जाता, ठीक है? So this could happen, ठीक है, randomly क्योंकि ये random assortment हो रही है. The same could happen here. ये वाला ऊपर आ जाता, ये वाला नीचे आ जाता, ठीक है? So random assortment, ये independent assortment metaphase में होती है. So it happens in metaphase 1 and metaphase 2 both, ठीक है? But crossing over only happened during prophase 1, right? So random assortment of sister chromatids during metaphase 2 results in production of genetically different gametes, right? So once the cytoplasm cleaves during the second cytokinesis, 4 haploid N daughter cells are produced. These cells are genetically different to each other and to the parent cell which began the cell cycle, right? So they are different, as you can see. Okay, so they can align randomly. Sorry, if you look at metaphase, they can align randomly. Okay, यहाँ पे आप see crossing over की वजह से genetic variation has taken place. तब जो भी daughter cell होगा बेटा, it will be completely different from its parent, and they will be different from each other also, right? So if you look at this graph here, so see, this is how it starts. The parent cell is diploid, remember. Okay? When cell cycle starts, remember, the cell during interphase at G1, okay, is the growth phase, right? During interphase, the S phase, the DNA will replicate. So you see from 2N, it has gone to 4N. ठीक है, जब वो 4N हो जाएगा, meiosis 1 start हो रही है. ठीक है, meiosis 1 we call it as a reduction division. By the end of meiosis 1, cytokinesis के बाद we will again have 2N, right? 2 daughter cells, 2N. ठीक है, अब वो meiosis 2 में जाएंगे. Remember, meiosis 1 के बाद directly meiosis 2 start हो जाएगी. There is no interface at all. So meiosis 2 खत्म होने के बाद cytokinesis के बाद we will eventually have 4 haploid daughter cells. ठीक है, so N. So this is how the nuclear division of meiosis takes place. Right? So meiosis produces daughter cells that are genetically different from each other. Daughter cells produced by meiosis are haploid. They are genetically different because one set of chromosomes comes from the maternal side, another set comes from the paternal side. Right? Crossing over of non-sister chromatids leads to recombination of genes. Also, independent assortment of chromosomes takes place in meiosis. So remember, this was taking place during metaphase 1 and 2. Crossing over was taking place during prophase 1. Therefore, daughter cells produced by meiosis are genetically different from each other. See, so different from their parents as well as different from each other, right? Now, so some distinguishing features which happen during each stage of meiosis 1. So something that you can actually use to review, right? So during prophase 1, remember, homologous pairs of chromosomes are visible. Okay, prophase 1, they become prominent. Metaphase, middle. Okay, remember it from middle. Homologous pairs are lined up side by side along the equator of the spindle. Anaphase, remember it from away. Whole chromosomes are being pulled to opposite poles with centromeres intact. Telophase, just remember it from 2. These are 2 groups of condensed chromosomes around which nuclei membranes are forming. Right? Then comes cytokinesis. Cytoplasm is dividing and cell membrane is pinching inwards to form 2 cells. Right? So these are the distinguishing features in meiosis 1. When we're talking about meiosis 2, prophase 2, remember, single whole chromosomes are visible. Okay, so this is the difference. Prophase 1 के अंदर we had homologous pairs. ठीक है, अब यहाँ पे आपके पास हो गया single whole chromosome. Metaphase में single whole chromosomes line up along the equator of the spindle. But remember, we talked about it will be at a 90-degree angle to the old spindle. Anaphase, again, remember it from away. Centromeres now divide. So this is the difference. ठीक है, वहाँ पे anaphase 1 में क्या हो रहा था? Whole chromosomes are being pulled. Centromeres are intact. Anaphase 2 में क्या हो रहा है? Centromeres are dividing. ठीक है, they divide and chromatids are now being pulled to the opposite poles. So उधर chromosomes were being pulled. यहाँ पे chromatids are being pulled. Telophase, the nuclei are forming around the 4 groups of condensed chromosomes. Cytokinesis, cytoplasm is dividing and 4 haploid cells are forming. Right, so something that you can use for a review for difference between meiosis 1 and meiosis 2. Right? Okay, so one question here. A student believes that she can use 2 raised to the power 23. So this is actually an expression that is used, right? It's 2 to the power of n, right? So a student believes that she can use 2 raised to the power 23 to calculate something related to meiosis. Now, if this calculation results in a number greater than 8 million, so you can actually work that out in your calculator, like 2 raised to the power 23, and it gives you a value, okay, which is more than 8 million, right? A value which is more than 8 million. Consider what she might be trying to calculate and why she chose that particular indices, right? So the thing with that is this is actually an expression, right? This expression is actually, remember, this is not for diploid. Diploid for the 2n. Okay, this is different. Okay, this is 2 raised to the power n. So this n actually tells you the number of homologous chromosome pairs. Okay, so when we're talking about a haploid cell, remember we have 23 pairs of chromosomes there, right? So ठीक है, नंबर ऑफ होमोलोगस क्रोमोसोम्स पेयर्स और हैप्लॉइड नंबर, ठीक है, एन 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 की बात कर रहे हैं, ठीक है, तो एन होता है 23 हैप्लॉइड के अंदर. So expression below can be used to calculate the possible number of different combinations of chromosomes following meiosis. ठीक है, so 2 raised to the power n is the expression. अगर इस n की जगह हम 23 लगाते तो you will get more than 8 million combinations. इतनी combinations हो सकती है. और remember अगर हम 23 chromosomes की बात कर रहे हैं one set की haploid की, तो more than 8 million combinations mother में है, more than 8 million combinations father के अंदर भी होंगी. ठीक है, और ये combinations आ रही है फिलहाल without crossing over. तो थिंक अबाउट, अगर क्रॉसिंग ओवर हो रही है तो उससे भी ज्यादा कॉम्बिनेशन बन रही है. ठीक है, नाउ देयर इज अनदर स्टूडेंट हु टेक्स दिस फॉर्मूला. ठीक है, उसने 2 raised to the power n का फॉर्मूला लिया. ठीक है, और शी डिड दैट 2 raised to the power 23, but then she comes up with a value of 7.04 into 10 to the power 13. What she has actually done here is that, like they are saying, what has that value come from? Discuss what the student has done to generate this value, how it can be used to add further detail to today's discussion about variation. So think about it. Remember, we're talking about haploid. So अगर एक paternal side से आ रहा है, using the same expression for the mother. So now if you multiply them, ठीक है, more than 8 million here, more than 8 million here, then you think about कि और कितनी आपके पास कितनी ज़्यादा combinations आ रही हैं आपके पास. ठीक है, so think about the genetic variations. Right? So 2 raised to the power 23 just a number. ठीक है, just the number of different possible gametes that one human can make. As sexual reproduction involves a random fusion of two gametes, potentially anyone from more than 8 million possible combinations that a male can make following meiosis can fertilize any one of the greater than 8 million possible combinations that a female could make. So think about what huge number of like different possibilities we have for gamete formation, right? So random fertilization of haploid gametes, right? This is another key event of variation. ठीक है, अब अगर gametes बन गए haploid, so they can randomly fertilize also. ठीक है, any male gamete can fuse with any female gamete, right? So random fertilization भी हो जाएगी. ठीक है, so this is just to actually recap what we have started talking about in meiosis. So we started this cell cycle with S phase. Remember, S phase was the phase of interphase during which DNA was replicating. So this is how the cell will look like. ठीक है, so centrosomes with the centriole pairs. ठीक है, अभी chromatin loosely coiled stage के अंदर है. ठीक है, तो अभी इस स्टेज में से DNA replicate हुआ है. जो already chromatin था वो replicate हो गया है. ठीक है, but they still remain as thin fine thread-like structure which is loosely coiled. So chromosomes will be duplicated during interphase. ठीक है, the resulting sister chromatids are held together the centromere, and the centrosomes are also duplicated, right? But still अभी भी thin हो गया. ठीक है, ये जो thickening और condensation होती है, it happens during prophase 1. ठीक है, अब now the nuclear division starts. So you can see chromosomes condense, so now they are more visible, they have become prominent, and the nuclear envelopes also fragments. ठीक है, homologous chromosomes, they bind firmly together along their length. So you can see homologous अब किनकी pairing up हो गई है. ठीक है, forming a tetrade. And अगर आप इस वाले को देखो, they are longer in length. So chiasmata has formed between two non-sister chromatids. Crossing over occurs at the chiasmata. Spindle fibers emerge from the centrosomes. So you can see ये centrosomes से spindle fibers से बनना शुरू हो गए. ठीक है, now metaphase में, so again better remember it from middle, but now they are aligning. So you can see कि अभी alignment शुरू हुई है इनकी. तो जरूरी नहीं कि दोनों अगर बायोवेलेंट थे वो एक साथ जाके कनेक्ट हो जाएंगे. तो एक के बाद एक, पहले एक लग रहा है फिर दूसरा लग रहा है, एक ऊपर एक नीचे है. So ठीक है, it will take time for them to arrange. So homologous chromosomes are attached to spindle microtubules at the fused kinetochore. So kinetochore are small portions of proteins that are found on the side of the centromeres, right? So they will attach to the spindle through those. ठीक है, and then what will happen? And these kinetochores, remember, these are like present on the centromeres, which are joining the two sister chromatids. Chromosomes continue to condense, and the nuclear envelope completely disappears. Right? Now, homologous chromosomes randomly assemble at the metaphase plate where they have been maneuvered into place by the microtubules. ठीक है, अब microtubules actually because the centromere से connect हो गए. ठीक है, but microtubules will actually because they can contract. ठीक है, they will pull them. ठीक है, and now they have aligned. Right? Anaphase में क्या हो रहा है? Spindle microtubules, they pull the homologous chromosomes apart. Right? The sister chromatid is still attached at the centromere. Remember, we're talking about meiosis 1. अभी ठीक है, जो आपके जो chromosomes थे, ठीक है, they are being pulled apart, or centromeres are still intact. Then sister chromatids arrive at the poles. So now you can see they arrive at the poles of the cell and begin to decondense. A nuclear envelope forms around each nucleus and the cytoplasm is divided by cleavage furrow. The result is two haploid cells. Each cell contains one duplicated copy of each homologous chromosome pair. So remember हमने 4n से start किया था और हम 2n पर आ गए दोबारा. Another thing that you might be noticing, क्योंकि जो crossing over हुई थी prophase 1 में and chiasmata was being formed, right? So they will only separate during anaphase, right? So उसी form में रहेंगे, ठीक है? chiasmata रहेगा metaphase के अंदर भी, anaphase 1 के अंदर actually they will be pulled apart, right? After that, telophase 1 and cytokinesis, the cell will undergo meiosis 2. Remember, no interphase will take place at this stage. Now we will start with prophase 2. So the sister chromatids now condense. A new spindle begins to form and the nuclear envelope starts to fragment, right? So you can see, right? Then the nuclear envelope totally disappears and the spindle fibers engage the individual kinetochores on the sister chromatids. Then again, independent assortment होगी यहाँ पे भी, ठीक है? So they can align randomly. Sister chromatids line up at the metaphase plate. So you can see here. Sister chromatids are pulled apart by the shortening of the kinetochore microtubules. Non-kinetochore microtubules lengthen the cell. So the ones which are attached here, ठीक है? तो ये तो microtubules इनको pull करेंगे, ठीक है? मगर जो non-kinetochore हैं, जो ये वाले हैं, ये cell को elongate करने में help करेंगे. These, ठीक है? तो ये cell को elongate करने में help करेंगे. So chromosomes that arrive at the opposite poles of the cell and decondense. Okay, this is happening during telophase 2. Nuclear envelopes surround the 4 nuclei. Cleavage furrow divides the 2 cells into 4 haploid cells. Right? So this is the whole summary of starting from interphase and meiosis 1 and meiosis 2. Right? So meiosis is a source of genetic variation. Right? So we have talked about these. Let's just recap. So it's having genetically different offspring. It can be advantageous for natural selection. Meiosis has several mechanisms that increase the genetic diversity of gametes produced. So we know about the mechanisms. One of them is crossing over that is taking place during prophase 1 and independent assortment or random orientation, which is taking place during metaphase 1 and metaphase 2. They result in different combinations of alleles in the gametes, right? So crossing over, okay, let's again review that. So crossing over is a process by which non-sister chromatids exchange alleles. So during meiosis 1, homologous chromosomes pair up and are in very close proximity to each other. The non-sister chromatids can cross over and get entangled. These crossing points are called chiasmata. The entangled places stress on the DNA molecules. As a result of this, a section of chromatid from one chromosome may break and rejoin with the chromatid from the other chromosome. The swapping of alleles is significant as it can result in a new combination of alleles on the 2 chromosomes. There is usually at least one, if not more, chiasmata present in each bivalent during meiosis. Crossing over is more likely to occur further down the chromosome away from the centromere. ठीक है, centromere से थोड़ा अलग होता है, ठीक है? But generally it happens, ठीक है, जहाँ पे अगर chromosome की length ज़्यादा लंबी है, वहाँ पे ज़्यादा crossing over के chances होते हैं, right? So you can look at the diagram here. See, so these are the homologous chromosomes, the bivalents, right? So this is sister chromatid, this is another sister chromatid coming from father, coming from mother, and these are the non-sister chromatids. So this is C, crossing over. Okay, so यहाँ पे see, you look at the length here. So this is what crossing over. इसकी वजह से ठीक है, there will be stress on the DNA, so they will break apart. Break होने के बाद क्या होगा? जो इसका जो ये end है, ये red इस पे आ गया और red वाले पे blue लग गया. So you can see here. ठीक है? So now you can see the different combinations here. Right, so exchange of genetic material takes place. So new cells that will be formed will have different alleles, right? And the new cells will have half a set of chromosomes from the mother, half a set from the father. Gametes are produced with different combinations. This is called independent assortment of chromosomes. क्योंकि prophase के बाद जब crossing over हो गई है, ठीक है? तो crossing over में finally they can then align differently also, right? So coming to independent assortment. Independent assortment is the production of different combinations of alleles in daughter cells during the random alignment of homologous pairs along the equator of a spindle during metaphase 1, right? And then different combination of chromosomes in daughter cells increase genetic variation between gametes. In prophase 1, homologous chromosomes pair up. In metaphase 1, they are pulled towards the equator of the spindle. Each pair can be arranged with each chromosome at the top, as we have already discussed in the diagrams. This is completely random. The orientation of one homologous pair is independent or unaffected by the orientation of the other pair. The homologous chromosomes are then separated and pulled apart to different poles. Combination of alleles that end up in each daughter cell depends on how the pair of homologous chromosomes were lined up, right? And then you can work out the number of different possible combinations, right? And you will be using the formula 2 raised to the power n, and where n corresponds to the number of chromosomes in a haploid cell, this n will be 23. So 2 raised to the power 23. So you can have more than 8 million combinations. Remember, this is for haploid cell. So if you calculate the same from the maternal side, then you can imagine we can have even more different combinations. So this is what they have actually shown you, these independent assortments. So remember, it happens in both, right? Meiosis 1, sorry, metaphase 1, metaphase 2, dono mein hoti hai. So here they have shown you 4 genetic arrangements that can happen. See, so this is आपको सेल दिखाया है मेटाफेज 1 के अंदर when it has aligned. ठीक है, it has aligned across the equator of the spindle. So here you can see the blue one is here, the red one is there. Then here we have the blue one and the red one. ठीक है, and if you look at the second combination here, blue and red इधर है, but अब यहाँ red आ गया और इधर blue आ गया. ठीक है, तो ये randomly अलग हो सकते हैं. ठीक है, हो सकता है पैटर्न इधर आ जाए, मैटर्नल इधर आ जाए. ठीक है, so see what happens. अब see metaphase 2 दिखाया है secondly. ठीक है, metaphase 2 में भी they will align. ठीक है, and now see here. After that, अब जब gametes बने हैं, so you look at, because ये वाला जो strand है and this strand, the smaller one, both of these will be pulled here. ठीक है, and this strand and this strand, both of them will be pulled here. So you can look at genetic arrangements. ठीक है, so these are 1, 2, 3, 4 haploid cells at the end. और इनको देख लें आप लोग, तो ये इनके कॉम्बिनेशन है. ठीक है, यहाँ पे क्या हुआ था, ये रेड वाला इधर गया, ये रेड वाला इधर गया. ठीक है, so they are together and now they will be pulled apart. सेकंड कॉम्बिनेशन में आप देख लें. ठीक है, अब हेयर क्या था, रेड इधर है और ब्लू इधर है. It was blue here and red there, and now look at the combination. This blue and this red. ठीक है, they have gone in one cell. This red and this blue, they have gone in another cell. ठीक है, अब metaphase 2 में again they align, and now look at the combination, blue and red, blue and red, and here red and blue, red and blue. So look at how many different genetic arrangements we can have. This is simply because of independent assortment, which happens during metaphase 1 and metaphase 2. Right? So just an example, several sources of genetic variation have been outlined above. It is also worth remembering that genetic variation can occur on even a smaller scale than chromosomes. So अभी तो हमने देखा कि chromosome में क्या क्या हो रहा है, crossing over हो रही है. Then we looked at independent assortment. But even then, mutations can occur within the genes as well. A random mutation that takes place during DNA replication, जब interphase में DNA replicate हो रहा है, उसमें भी mutations हो सकती हैं. जो sequence है nitrogen bases का, वो change हो सकता है. Okay, it can lead to production of new alleles and increased genetic variation. Okay, so this is all what will lead to daughter cells which are totally different from each other as well as from the parents. And with this we end the whole process of meiosis. And now we look at a little short animation of this whole process.
Have you ever wondered why children do not look exactly like their parents? Or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis 1 and meiosis 2. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase 1 of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase 1, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar, but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase 1 concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers forming the meiotic spindle. In prometaphase 1, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase 1, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase 1, the homologous chromosomes separate and move toward opposite poles. Meiosis 1 ends with telophase 1, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the 2 daughter cells are separated by a cleavage furrow. Since the final product of meiosis 1 is 2 haploid cells, meiosis 2 begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase 2, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase 2, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase 2. During anaphase 2, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase 2, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs and cleavage furrows separate the 2 daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate. and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.